thank you very much, and I'm very pleased to be presenting here for the first time. Inotek is focused on glaucoma, and you've seen several presentations on glaucoma today. Clearly, the optimal drug, eyedrop, to treat glaucoma would be an eyedrop that both preserves the IOP or lowers the IOP in a well-tolerated fashion, but also is neuroprotective. And currently, all treatments lower IOP as opposed to being neuroprotective. And the primary outflow mechanism in lowering IOP is the trabecular meshwork. And yet, many of the existing drugs and treatments serve to divert the aqueous outflow from the trabecular meshwork. So our approach is to restore the function of the trabecular meshwork in IOP reduction. The eye drops that are used for IOP reduction have been around for a very long time. The prostaglandins were launched in the 1990s, and the second-line therapies are even older than that. And the efficacy of these drugs creates a real need. About 50% of patients that start on a first-line therapy require second-line treatment. So the efficacy is suboptimal. And you're also combining different dosage regimens. Some of them are QD, some are BID, and some are TID, which causes issues with compliance. The other real issue with compliance is the side effects of many of the existing treatments. The prostaglandins in particular can cause severe hyperemia, which is implicated heavily in the lack of compliance associated with these drugs. And some of the second-line agents can also cause some severe systemic effects, which in certain cases can be very severe. So clearly, there's a need for a drug that is well-tolerated and is also very effective. Inotek's focus is on adenosine, and we've been working on adenosine for many years. We started out looking at it in ischemia models of stroke and heart attack, and we ended up working on it in the eye. And we focused in on one specific receptor of the 4 subreceptors that are associated with adenosine. So we created a mimetic that is very specific to the A1 subreceptor. And what we needed was a drug that was very potent so that we could put enough in one eye drop to have good efficacy. We have a drug that is very selective. You can see from the K1s the KIs at the bottom of this slide that we're very selective for the A1 subreceptor. We need a drug that penetrates the cornea effectively. And fourthly, we need a drug that is well tolerated, which is really one of the unmet needs in this market. So why the A1 subreceptor? That work was not done by us. It was done by Dr. Mort Seiven at the Shea Eye Institute some years ago, when he put out a study that showed that if you stimulate the A1 subreceptor, you reduce IOP, whereas if you stimulate the other subreceptors of adenosine, you actually increase IOP. So that got us very focused on the A1 subreceptor, and that's why we're very specific for that subreceptor. Trabadenosin's mechanism of action is to clear the outflow pathway through the trabecular meshwork. The trabecular meshwork represents about 70% of the outflow pathway, while the uveoscleral outflow represents the other 30%. And the trabecular meshwork in a glaucomatous eye becomes clogged with proteins. And trabadenosin acts by upregulating MMP2, a protease, which clears out the proteins and restores the natural pressure control through the clearing of the proteins in the trabecular meshwork. So our first study, of course, was a phase 1 study, and we wanted to test how high we could go on the dose to get a maximum tolerated dose. And what we did was we took 70 adult healthy volunteers, and we gave them 6 different escalating doses in one eye. And it was very well tolerated. We subjected these patients to Holter monitors, telemetry. We were measuring cardiac function. We were measuring eye function. We gave them sleep scales. So we were really measuring everything we could think of to measure. And we didn't see any any issues with 3,200 micrograms in one eye. So we amended the study and put 3,200 micrograms in each eye, and we still didn't see any side effects associated with that. So we think that what's going on the systemic side is that there's a saturation of the tissue. So as you go up in dose, you're not actually systemically absorbing any more drug. So then we started our phase 2 program, and we started that, as you can see from this study, we've shown that as we double the dose, we get about a 0.7 to 0.9 millimeter increase in IOP reduction with each successive doubling of dose. The first study was a monotherapy study in the same patient group that we're going to be testing in phase 3, patients with IOP of 24 millimeters or above, not on drug. And we found that we had very good dose escalation, going from 50 to 500 micrograms, and the efficacy didn't seem to be topping out at all at 500 micrograms. So there's clearly still the ability to go to a higher dose. And then we took the 500 microgram dose, and we took it from 2 weeks. This was initially a 2-week study. We took it from 2 weeks to 4 weeks, and we saw a statistically significant improvement in efficacy at 500 micrograms between 2 weeks and 4 weeks. So we showed that the efficacy of the drug in that period is actually getting better over time, which is important because several drugs in this category have tachyphylaxis. So then we decided to work on the patients that we would test in our fixed-dose combination. And we tested it in PGA poor responders, so patients with IOPs of 24 or above that were on PGs. And as you can see, we saw that when we went from 500 micrograms BID to 1,000 micrograms QD, we again got a very good increase in IOP reduction. So that set us up for our phase 3. We're starting our first phase 3 monotherapy trial. We're testing doses of 1,000 and 2,000. So these are diurnal results. We're testing doses that are much higher than the ones we tested in phase 2. So onto the back of the eye. Clearly, there's a need there to protect the retinal ganglion cells. And what we've shown in rat models is that our drug gets to the back of the eye, and there are A1 subreceptors there. In a high-pressure model, we can preserve the retinal ganglion cells, and we've also shown that we can get drug to the back of the eye in rabbits and monkeys. So we're excited to that, and I appreciate the opportunity to present at OIS. Thank you.